morning, students of class 11. Welcome to today's biology class. Students, we are studying the chapter Cell, the Unit of Life, is it not? So before starting with today's topic, let us recapitulate what we have learned from this chapter in the past few classes. So we started this chapter studying about cell theory, which was proposed by Schleiden, Swan and Rudolf Virchow. The statements of the cell theory were that all living organisms are composed of cell and products of cell, and all cells, they have arisen from a previously existing cell. Then we got to know about the overview of the cell, the different kinds of plants and animal cells. The largest cell, the ostrich egg, to the smallest cell in human body, the female egg. Then the cells of different shape. We came to know that the cells, they have a variety of shape based on the function they do, right? Then we started studying about prokaryotic cells. We came to know that these prokaryotic cells are the cells which do not have membrane bound nucleus. These cells also lack compartmentalization, and so they do not have any cell organelles. This cells, or these prokaryotic cells, they include organisms like bacteria, pleuropneumonia like organisms, mycoplasma, etc. And they exist in a great diversity of shapes and sizes. Now in the prokaryotic cell, we studied about the different parts. Like we started studying about the cell envelope, which is made up of three layers: the glycocalyx, cell wall and plasma membrane. We came to know that glycocalyx is the outermost layer and it is quite thick. This glycocalyx can be of two different types. Sometimes in some bacteria is slimy, and in some bacteria is quite thick and tough. We call it a capsule, right? Below the cell envelope there is the middle layer, below the glycocalyx there is the cell wall, right? And this cell wall is very important to give the structure and shape to the prokaryotic cells. Then finally, the innermost is the plasma membrane, and plasma membrane is semi-permeable and it controls entry and exit of different molecules. Like plasma membrane is same in all kind of cells. Then we studied about the different types of cell envelopes. Some cell envelopes are very tough and some are slimy. So based on the type of cell envelopes and their response to Gram's staining process, the bacteria are divided into two types: gram-positive bacteria that take the Gram stain, and the gram-negative bacteria that do not take the Gram stain. You can see in the image, this person, Christian Gram, he developed this staining method. And the Escherichia coli is a gram-negative bacteria that doesn't take this Gram stain. And Staphylococcus is a gram-positive bacteria. So this technique can be applied to identify what kind of bacteria is in our body, in the blood, etc. Then we studied about mesosome. Mesosomes are some infoldings in the form of vesicles, tubules, lamella, etc. These mesosomes have got a wide variety of function, like formation of cell wall, replication, for helping during formation of the daughter cells. They also increases the surface area of the cell. They help in respiration, secretion, etc. Wide variety. This mesosome, I will tell you that these are actually the replacement of the cell organelles. Prokaryotic cells do not have cell organelles, so mesosomes are just a replacement of it. Some of the mesosomes, they are laden with pigment containing molecules. They are called as the chromatophores. So chromatophores are taking the function of the plastids. Say in case of the bacteria, cyanobacteria, blue-green algae, they have got pigments, but there is no plastid, no organelle. So these pigments are there in the chromatophore in the form of some pigment containing molecules. You can see in that image. Then we studied about the nucleoid. As the prokaryotic cell do not have a membrane-bound nucleus, then the genomic material is present in an area that is called as a nucleoid. The DNA appears to be in form of fibers, an arrangement of fibers, Apart from the nucleoid, that is the chromosomal DNA, most of the bacteria have an extra chromosomal circular autonomously replicating DNA that is called as plasmid, and we came to know how plasmid gives antibiotic resistance to prokaryotic cell and its application in antibiotics. Just a gist we got to know about the application. Then we came to know about two more structural parts of prokaryotic cell. The flagella. Flagella helps in movement of the prokaryotic cell. Then pili and fimbriae. Pili and fimbriae are very short, fine hair-like extensions. This pili and fimbriae, they will help in binding the bacteria to any substratum or to the another bacteria. So in the image you can see the fimbriae, which will help the bacteria to stick to substratum, and there are some pilas. Pilas will help, singular form, pilas will help the bacteria to bind to any other pilas of a bacteria, different bacteria. Then finally, yesterday's class we studied about the ribosomes. The prokaryotic cells, they have got a different ribosome. The ribosome is of what? 70S, and we came to know that these ribosomes, they are having two subunits, the larger subunit and the smaller subunit, and these ribosomes are not dispersed in the cytoplasm, but they are bound to one mRNA. You can see in that image the mRNA. In the mRNA, the green color, the mRNA has got so many ribosomes stick. So this structure of mRNA stick with so many ribosomes is called as polysomes or polyribosome. This is very important. Polyribosomes are important because polyribosome is the structure which is producing protein from the mRNA, right? And then finally we studied about the inclusion bodies. So inclusion bodies are some organelles who stores or who reserves the material in prokaryotic cells, like the food granules, etc. So this much we studied about the prokaryotic cell. So in this class, what we have got to know. Let us see. Today's the learning outcomes will be to list the basic characteristics of eukaryotic cell. We'll not go in detail in today's class. We just go to the basic characteristic. Next, we will name the cell organelles of plant and animal cells. We are going to describe the structure of plasma membrane according to the fluid mosaic model. So let us get started. Eukaryotic cells. Already we have known so many characteristics about eukaryotic cells from class 9 onwards. So eukaryotic cell, already we know that these cells have gotten well organized membrane bound nucleus, as well as they have got membrane bound organelle. So presence of this membrane gives a clear compartmentalization of the cytoplasm. The cytoplasm has got so many organelles, and these organelles are like the compartments inside the eukaryotic cell. Another very important characteristic is that the DNA in eukaryotic cell do not appear like fibers what we have seen in the prokaryotic cell. They appear as chromosomes, some rod-like structures, not as fibers, some rod-like structure called as chromosome inside the nucleus. Then cell organelles. What are the cell organelles in a eukaryotic cell? Eukaryotic cells have got large number of cell organelles which are not present in prokaryotes. So in a plant cell, let us see what are the cell organelles. Cell membrane, the outermost. Cell membrane is present, or plasma membrane. Cell wall is there. Then endomembrane system is there, and this endomembrane system consists of endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles. So we'll go into all these organelles in detail in the coming classes. This is just a beginning of eukaryotic cell. Another very important organ is the mitochondria, the powerhouse of the cell. The plant cells also have plastid. Plant cell also have got ribosome like prokaryotic cell. Plant cells have got cytoskeleton. The cells have got also nucleus and microbodies. Now let us take an animal cell. Animal cells, they do not have cell wall. They have just the cell membrane or the plasma membrane at the outside. This cell animal cell also have endomembrane system consisting of endoplasmic reticulum, Golgi apparatus, lysosome and vacuoles. The animal cell will have mitochondria, animal cell will have ribosome, cytoskeleton, and a very important organ which is not present in the plant cell, that is centrosome and centrioles. Apart from this, eukaryotic cell of course will have a well-defined nucleus, will have microbodies, right? But this animal cell, it do not have the plastid like plant cell. Now, the first structure we will study in detail in eukaryotic cell is the cell membrane or plasma membrane. So, if you see in the image, this is a schematic diagram of a plasma membrane. We know plasma membrane is present in both plant and animal cell, right? And this plasma membrane is made up of chemicals, of course. But which chemicals? Which chemicals? The plasma membrane bounding every cell is made up of two lipid layers. If you can see in the image, those those blue colored circles, these are two in layer and these are bilayer, that means, and these are lipid molecules if you can see properly. Apart from this bilayer lipid, the plasma membrane or cell membrane also consists of proteins. These proteins, if you see properly, some of the proteins are just floating above. They are not penetrating into the lipid layer. The proteins, which are the protein molecules, large size protein molecules, which are just floating above, okay, will be called as the external proteins. And these proteins which are deep into, they are transcending the, they are passing through the lipid molecules, they will be called as the integral proteins. Okay. Now, this lipids, the lipids mainly, they are phosphoglycerides. Lipids are of different kind. In the coming chapters, we will understand. Lipids are like oil. But which kind of lipid? This lipid which is present in the plasma membrane is a phosphoglyceride. That means along with the lipid molecule, it will also have the phosphate molecule. And will have a polar head and an inner hydrophobic tail. We'll discuss all this in detail in the coming classes. Okay. Now, this protein and the lipid ratio in different cells are not same. Like if we take the example of human RBC, the amount of protein is very high. Like take the example of this RBC. In the RBC, the percentage of the protein is 52%. Whereas for lipid it is 8% and the 40%, and the remaining 8% is the carbohydrate. So we come to know that the plasma membrane or cell membrane consists of three biomolecules. The lipid, that means the phosphoglyceride particularly, then proteins of two different kinds, and also carbohydrate, the least in amount. Already I told you that these proteins are of two different types. The integral, which totally are buried or partially buried in the membrane, which pass through the membrane, is the integral protein. And number two is the peripheral protein, which just float at the surface of the lipid bilayer. Okay. So, this membrane structure, whatever is we know today, was put forwarded by these two persons which are in the screen. Okay. Fluid mosaic model given by Singer and Nicholson. So they give a very remarkable model. This fluid mosaic model helped in the field of coming research of biology tremendously. According to this model, whatever the model they gave, the plasma membrane, they show a quasi-fluid nature. Quasi-fluid means the plasma membrane is always floating. That's why we call it a living membrane. And it also allows lateral movement of the protein over the lipid bilayer. Lipids are also floating, but among them the proteins are also floating. So we can compare this plasma membrane as like an ocean, and this lipid molecules are the water of the ocean, and the protein are the icebergs. So like how icebergs float in the ocean, In the same way, the protein molecules, large globular protein molecules also float in the ocean of lipid molecules. So the ability of this movement of the ability of movement of this lipid molecule is the fluidity of the plasma membrane. The fluidity of every plasma membrane is not same, and we can increase and decrease also. In coming classes of biology we'll come to know, right? So now come to the functions of plasma membrane. Plasma membrane or cell membrane is so important for all the living organisms. The main function is the control of transport of molecules. This plasma membrane is selectively permeable. Why? Because it allows only selected molecule to pass to and fro, to enter and to go out. Due to this quasi-fluid, the fluidic nature of this plasma membrane, it is possible for a cell to grow. It is possible for a cell to engulf. Take the example of amoeba. For this fluidic nature, it is possible for a cell to divide. A mother cell can divide into two daughter cells. Okay. Because of this plasma membrane, there can be formation of the junction which we have studied, like three kind of junctions: tight junction, adhesion junctions. So this is the specialty of the plasma membrane. Plasma membrane is very important. It is not only important because it's the outermost covering layer, also because it is because of its nature, because of fluidic nature. So this fluid mosaic model is very important in the history of biology, and also in the exam purpose also of the class 11 syllabus. So let us recapitulate at the end what we have studied in today's class. We started studying eukaryotic cells, and we came to know that eukaryotic cells have got compartmentalization. They have got cell organelles. They have got a well-defined nucleus, and inside the nucleus, the genetic material, the DNA, is packed into chromosome. It is not in form of fiber, network of fibers like prokaryotic cells. We came to know that the animal cells, they do not have plastids. Is it not? And plant cells, they do not have the centrosome and centrioles. Animal cell do not have plastid is very common, but plant cell do not have centriole is maybe new to you all in class 11. So pay attention. Then we came to know about plasma membrane. We came to know that plasma membrane is made up of three biomolecules: lipid, which is arranged in two bilayers, then proteins, which are of again two types, peripheral and integral, and also carbohydrate in a minimum amount. Plasma membrane has a wide variety of function. Plasma membrane is selectively permeable. Plasma membrane helps in endocytosis, in engulfing. Plasma membrane helps in division of the cell, helps in growth of the cell, in multiplication of the cells also. So this much for today's class. Please watch the video more than once if you have some kind of doubt. If still you have any kind of doubt, you can ask me, and your homework will be uploaded in a Google Classroom. Please submit. Please do that, complete it and submit on time. Okay. So thank you for attending the class.